Welcome geniuses, I'm Genie, your best buddy for A-levels. In this channel, we'll bring you to explore the secret formula behind success.
For this video, I'm going to cover topic 4, transport across membrane, where I'm going to focus on the movement of substance in and out of cells, and I'm going to introduce you with all the different types of transport, such as diffusion, facilitated diffusion, osmosis, active transport, endocytosis, and exocytosis. So we're going to see the relationship between all of this and how to differentiate the different types of transport. So I'm going to start the introduction about the different types of transport across membrane. Generally, we have two categories of the type of transport. We have active and passive. So what is the difference between active and passive? As you know, passive means it's a process that does not require ATP, whereas active process requires ATP. So what are the processes that doesn't require ATP and why they don't need ATP for the movement? Because the molecules move down concentration gradient. So what are the examples of process that is under passive transport? Total, we have 3. The first one will be simple diffusion. Simple diffusion. What kind of molecules uses simple diffusion? We have molecules that are small, uncharged, non-polar, so they can pass through the phospholipid bilayer hydrophobic core. Examples, oxygen and carbon dioxide. All lipid-soluble substances. Another type of transport, we have osmosis. Osmosis is actually similar to diffusion. It's just that it's exclusively meant for water molecules. And the movement of water molecule is down water potential gradient. We don't use the word concentration. Through a partially permeable membrane. And the last passive transport will be facilitated diffusion. So we have two types of facilitated diffusion, such as channel protein and carrier protein. So what's the difference? The shape is different and the type of molecule they transport is also different. Channel protein looks like this, where it has a hydrophilic pore, whereas carrier protein actually changes shape and it has a specific binding site. So this is the channel protein, while the other one will be the carrier protein. So what is the difference? Channel protein transport small molecules and usually charged, such as ions and water molecules, whereas carrier protein transport usually large and polar molecules. Example, glucose, amino acid. There's another type of facilitated diffusion known as co-transporter, where in topic 7, transport in plants, you will come across this type of co-transporter where you need two types of molecule to be transported together. And usually, these molecules are of different types, where one of the molecule will be moving down the concentration gradient, whereas the other one will be moving against the concentration gradient. That's why I say it's a very special kind of transport, because despite the second molecule is against concentration gradient, it does not require ATP. So it's under passive transport. Now I'm moving on to active transport. So active transport generally requires ATP, but not every transport moves the molecule against the gradient. One of it is known as active transport, whereas the other one is known as active process. So the difference is, one requires carrier protein, while the other one does not. So active transport requires a carrier protein, and molecules are moved against concentration gradient. Example, you would have seen sodium-potassium pump, where the ions are moving against concentration gradient. For active process, it is also known as bulk transport. So the membrane can take in or release large molecule. So this is for large molecules. So the process of taking in will be known as endocytosis. Whereas releasing will be exocytosis. Under endocytosis, there are two different ways. One is taking in solid and one is taking in liquid. So the one taking in solid is known as phagocytosis, whereas the liquid is known as pinocytosis. For more details and understanding for this chapter, you're welcome to join my lesson.
That's all for today's video. If you're interested in more genuine sharing by other geniuses, please subscribe to our channel and don't forget to turn on the notification bell, ding dong. Also, if you're struggling with one or two past year questions and the mark scheme just doesn't seem to help, Genie has got you covered. Feel free to let us know what question it is by filling in the Google form linked in the description below. GenieUs Hub will get genius teachers to fulfill your request for the solution. Genie will see you next time. Bye-bye.